Hello everyone and welcome to today's webinar. We're excited to have you with us as we explore effective strategies for streamlining manufacturing and analytics in cell and gene therapy development. Together we will tackle the critical challenges that can hinder the path to a commercially viable CGT product. But before we dive into today's webinar, let me share a quick overview of what we will be covering. My name is Laura and I'm the Group Leader for CGT Analytics Applications at Miltenyi Biotec. I will start with a quick introduction to our company, followed by an outline of our platform dedicated to streamlining cell manufacturing and analytics. Then my colleague Silvio will delve into strategies for enhancing efficiency while maintaining flexibility. And lastly, my colleague Dirk from Miltenyi Bioindustry will present our comprehensive end-to-end support for Lentiviral vector and CGT product development. For over 3 decades, we have been at the forefront of scientific innovation, developing technologies, instruments, reagents, and services that have propelled both our company and the field of CGT forward. And thus, each year, over 10,000 patients benefit from treatments made possible by our technologies. To better support the CGT community, we have established subsidiaries and branch offices across the globe. For example, in the United States, we have locations on both the East and the West Coast, as well as 2 Clinimax cell factories in Gaithersburg and San Jose, where we offer services for lentiviral vector design and manufacturing, along with process development and analytical development. Additionally, last year we proudly opened our first direct office in India. Miltenyi is actually comprised of 3 distinct divisions, including Miltenyi Biotec, Miltenyi Bioindustry, and Miltenyi Biomedicine. At Miltenyi Biotec, we focus on developing innovative tools and providing comprehensive customer support and services designed to facilitate the translation of your biomedical advancements into clinical applications. Our Miltenyi Bioindustry side represents our globally fully integrated CDMO organization that supports you at any step of your development process. And our Miltenyi Biomedicine is committed to bring cancer treatments and regenerative therapies to patients worldwide by developing personalized CGTs. The field of CGT is rapidly evolving, and with it the pipeline to provide new innovative treatments for patients worldwide. However, as we all know, the path from clinical development to market approval remains challenging. Manual and fragmented workflows, non-commercial ready platforms, non-compliant software, lack of data integrity, limited technical and regulatory support are just a few of the hurdles CGT developers must overcome. To speed up your CGT journey from early discovery to commercialization, we provide comprehensive solutions. Next to cutting-edge technologies such as the Clinimax Prodigy and the MaxQuant Analyzer, we offer support and services for various applications like, for example, engineered T cells, NK cells, PSCs, and many more. Moreover, we offer instrument services and training courses to make you an expert user of our products in no time. In addition, our regulatory support team helps you navigate requirements from clinical trial submission to market approval. When selecting a platform for CGT manufacturing and analytics, it's essential to prioritize early automation, seamless integration, and scalability to ensure you reach your milestones sooner and smoothly transition from development to commercial phase. To empower you, we have actually built a proven platform where manufacturing meets analytics. Or in other words, where the Clinimax Prodigy pairs with the MaxQuant Analyzer. The perfect match, if you will. On the manufacturing side, we have our Clinimax Prodigy platform, the flagship product and reference standard for integrated cell processing and manufacturing. As an automated and closed platform, the Clinimax Prodigy integrates all essential processing steps from cell washing to final formulation into one efficient application. It's a versatile all-in-one solution for both autologous and allogeneic cell therapy manufacturing. Moreover, thanks to the flexibility of the CliniMax Prodigy platform, you can find it all the way from process development to commercial, as it easily adapts to your scalability needs. So no surprise, it's the #1 platform for automated cell manufacturing and used in over 200 active clinical trials, hospital treatments, or commercial treatments around the world. On the analytics front, we offer the MaxQuant Analyzer, compact benchtop flow cytometer designed with advanced automation features. From routine instrument maintenance and acquisition to automated data backup and analysis, the MaxQuant Analyzer streamlines your workflow for maximum standardization and efficiency. The integrated software further allows you to be audit-ready due to the 21 CFR Part 11 module for regulatory compliancy, and our SmartGain technology facilitates inter- and intra-instrument alignment for reproducible performance across sites. Via our express modes, automated analysis and reporting is available at the push of a button directly on the instrument. And our MaxQuant analyzers are already authority approved as they are included in several clinical trials. Whatever application you're running on the CliniMax Prodigy, analytical testing is tightly connected to the manufacturing process and goes hand in hand for in-process control, release testing, and concomitant research. In-process control is done at key intermediate stages during manufacturing to monitor and, if appropriate, to adjust the process to ensure that the final product conforms consistently and reliable to its specification. Release testing of the final drug product confirms alignment with established release criteria. And during concomitant research, analytical tests are in general conducted to understand the biological mechanism and to predict the clinical outcomes. To better understand how the two instruments work together in one integrated workflow, let's have a closer look at a specific example, namely the CAR-T or TCT process. This process depicted here on the left begins with your starting material, like the leukapheresis product, and in multiple steps the target cells are then separated, modified, expanded, and finally formulated into the drug product on the CliniMax Prodigy. And at key intermediate stages during the manufacturing, analytical tests are performed to determine, for instance, the whole blood cell count, cellular composition, viability, and transduction efficiency. And these in-process controls are generally done by flow cytometry as depicted here with the green color. Additional tests are also done like mycoplasma and microbiological examination to further confirm the sterility at important points. Let's zoom into one specific step shown here, where the cell labeling and separation is performed. For this, the CliniMax Prodigy asks for specific parameters like the white blood cell count, frequency of labeled cells, and the cell product volume. And for this, you can just take a sample and directly hand it over to your analytical or QC team. And all they need is a MaxQuant analyzer, a respective express mode, and the Stain Express immune cell composition cocktail to easily determine the needed parameters and to continue the process. After running your in-process control, you can take those numbers shown on the express mode and take it directly back to your operator at the CliniMax Prodigy, which then enters the required information to continue with the processing step. Previous example nicely demonstrated the synergy between the two instruments and highlighted the advantages of our co-developed harmonized workflows for manufacturing and analytics. But now in more detail, what are actually express modes? Well, express mode, as you can see here, are unique add-on features for the MaxQuantify software. Developed to simplify flow cytometric analysis, they provide full software automation via predefined experiment settings, acquisition, and automated analysis. Thus, the usage of express modes reduces errors and ensures reliable and reproducible results. In addition to our pre-installed and add-on express modes for various applications such as CAR-T and NK cells, we also provide custom express modes that are specifically tailored to meet your analytical requirements. Moreover, we offer a huge portfolio of reliable and precise reagents for flow cytometry, like our affinity recombinant antibodies, or shown here our stain-express dry antibody cocktails. These are pre-formulated dry antibody cocktails designed so you don't have to worry about panel design, cocktail preparation, pipetting inaccuracy, human error, refrigerated storage, stock planning, or manual analysis. All you need to do is to add your sample, and with the respective express mode, you get reliable results at the push of a button. Next to the Stain Express Immune Cell Composition cocktail, we offer dried cocktails for CAR-T transduction efficiency, CD19 CAR-T monitoring, and for T-cell subset analysis. And with that, I hope I could give you a brief overview of our integrated platform solution for manufacturing and analytics. So let this perfect match realize your CGT vision and take advantage of streamlined workflows that reduce manual intervention, enhance operational efficiency, and maximize reliability. The commercial readiness of our proven platform that ensures effortless scalability and seamless technology transfer. Custom solutions that are tailored to your specific needs and CGT applications, decades of expertise and dedicated support, along with optimized consumables and reagents, and all of this provided by one trusted partner in CGT. Additionally, we are here to support you at every step of the way to accelerate your CGT program. Our service contracts encompasses maintenance, repair, support, and qualification services, ensuring the long-term performance and reliability of your instruments. As mentioned earlier, our dedicated custom application service team can customize any cell processing or analytical step to your needs. Moreover, we offer various courses on the newest techniques, applications, or technologies specifically designed for you. Whether you prefer live webinars, interactive tutorials, or hands-on training led by our experts at one of our Miltenyi Innovation and Training Centers, or even at your own location, you can easily find the right option to enhance your learning experience. Lastly, we are also here to assist you with any regulatory questions you may have. And with that, I'm at the end of the first part and I'll hand it over to Silvio.
Now it's time to explore in more detail how Miltenyi's platform technology can help you to leverage the full potential of cell and gene therapy manufacturing and analytics. As you have heard in the introduction, the CliniMax Prodigy platform is a very flexible, integrated, closed, and even customizable manufacturing platform. All of these attributes have been introduced by design and have been proven in thousands of preclinical, clinical, and most importantly, also commercial cell and gene therapy manufacturing procedures. In more detail, the CliniMax Prodigy has shown its versatility in more than 180 clinical trials. A plethora of varying cell and gene therapy companies and institutes, more than at least 45 different countries, and with no less than 300 customizable applications, which have been developed on top of the more than 20 pre-installed standard cell and gene therapy applications on this device. What is the secret behind all of these astounding figures? It's for sure a very solid understanding of the underlying biology of cell and gene therapy and a strong intention to bring this sophisticated technology into a commercially viable product, which is ready for industrialization on a very large scale. At the very first glance, the CliniMax Prodigy manufacturing platform might look quite complicated, but when you begin to have a closer look at the different modules of the platform, things become far more intuitive and simpler than you thought. In a nutshell, the CliniMax Prodigy platform consists of a seamless interplay between hardware, software, disposables, and consumables. Let's walk step by step through the different modules. At the heart of the CliniMax Prodigy platform comes the centrical unit, which is able to incubate cells at a defined temperature and gassing scheme. Pellet cells with varying speeds and intensity and feed or exchange buffer media or any other ingredients without the need to remove the cells from the controlled cell cultivation environment. Prior to the cell cultivation, the magnet unit will allow for selection and/or depletion of your cells of choice. The process is fully automated and closed, and the different unit steps can be followed via the graphical user interface. The peristaltic pump allows for gentle transfer of cells through the tubing set, which has been designed and optimized as one system, together with the application software and a toolbox of reagents, buffer, and media. Process inputs and outputs can be attached and detached via accepted aseptic procedures, such as welding, and can be traced via integrated barcode reading and subsequent data transfer to the process log. The highest degree of flexibilization on the CliniMax Prodigy platform is given by two different layers of software. The primary layer is the operating system, also called core software, which is the foundation of all standard and customized applications. As an end user, you are mainly interacting with the application software layer, which is operated via a graphical user interface that can be seen on this slide. As already mentioned before, more than 20 pre-installed standard applications, plus another at least 300 customized application programs demonstrate the immense flexibility of this system. On top, any information which needs to be communicated from the CliniMax Prodigy to another machine or a conventional paper-based record, executed by an operator and revised by a qualified person, is orchestrated via the operational system. The most recent version of the operational system will give you full 21 CFR Part 11 compliance and pave the way to full IT connectivity in the near future. Talking about connectivity, the closed nature of the CliniMax Prodigy platform is another key feature of the CliniMax Prodigy platform. Sterile welding is still the preferred way to connect process inputs and outputs. The design of the tubing set, as well as the selection of additional accessories, allow the user to achieve aseptic connectivity of virtually all process steps directly at the manufacturing platform, enabling to perform the manufacturing procedure in a Class 7/ISO 7 cleanroom or even lower. When looking at the currently commercialized cell and gene therapies, most of them do still use manual and partially open formulation and fill operations. In order to overcome this bottleneck, we have further closed the CliniMax Prodigy manufacturing platform via a fully integrated formulation and fill module, which comes with virtually no addition to the manufacturing footprint and allows for 100% interoperability and interconnectedness to the manufacturing platform. The 13-valve concept leaves you with significant flexibility with respect to drug product, QC, and sampling. Equally to the already fully automated upstream manufacturing standard applications, the formulation and fill device comes as a system development product, containing of the previously introduced hardware, a specifically designed disposable, and in most cases, fully customized formulation and fill applications. The current composition of the formulation set is given by 4 larger product bags for up to 500 ml of freshly filled cell suspension, and 4 smaller product or QC bags for up to 50 ml of freshly filled cell suspension, and a waste bag for initial priming activities and subsequent unit operations to achieve highest cellular homogeneity between the different bags. It is possible to freeze all of these bag sizes, albeit allowing for smaller validated fill volumes. Further modifications at the dedicated formulation set are possible via welding operations. Very recently, we released the CliniMax workbench, which allows for seamless integration into Miltenyi's manufacturing ballroom concept. We will hear some more information about this concept from our next speaker, Dirk Windgassen. The CliniMax workbench has quite some GMP-relevant features and benefits, like uninterrupted power supply, central supply connections via sealing sockets, a severely reduced manufacturing footprint per device, and smart cleaning and maintenance supporting design. Most importantly, rearrangement of the CliniMax Prodigy device does not need requalification in case it's moved via the CliniMax workbench. Talking about system design, Miltenyi's so-called standard applications come together with the instrument's operational system and do not need any further installation or additional software verification. Despite the inbuilt GMP readiness, most of the standard applications offer a high degree of flexibilization, especially when they are used in combination with Miltenyi's reagent, buffer, and media toolkits. Most importantly, the application layer is the prerequisite to achieve a perfect match with the analytical procedures and the connection to the max quant. It has been designed with the knowledge in mind of how to develop and run IPC/QC operations in cell and gene therapy manufacturing. Miltenyi's T-cell transduction is the powerhouse workflow for engineered T-cells in the cell and gene therapy space. Every year, thousands of runs are conducted using the depicted workflow, which is based on the standard application software T-cell transduction, or short TCT. As you can see on the very right, it can be easily combined with certain analytical workflows, including sophisticated automation procedures via reagents, software, and even hardware solutions. You have already heard about the possibility to rapidly stain your cells with Miltenyi's freeze-dried antibody cocktails called Stain Express, and subsequently analyze them with automated express modes to minimize hand-on times and reagent usage during analytical procedures. For all major unit operations, such as cell selection, cell activation, cell transfection or transduction, cell expansion and harvest, tailor-made reagents are available to achieve a highly scalable and robust manufacturing process. We have set up the system for cellular expansion times of 8 to 14 days. But of course, it's technically possible to further shorten or prolong your process, depending on sufficient process qualification. Due to the centrifugal size, the total potential amount of, for example, CAR T cells is limited towards a maximum of roughly 7 to 8 times 10 to the 9 cells, which is usually fully sufficient for CAR T cell manufacturing. However, when needing more cells, Miltenyi also offers a larger chamber integrated in a new tubing set called TSX20 and coming with a so-called TCT large scale, in short TCT-LS standard application, having also adapted activation reagent to be able to seed and activate more cells at the beginning of the process. On the last slide, I shortly mentioned that the TCT also follows for a very good match with Miltenyi's analytical process portfolio. Here you can see that taking samples and analyzing them via flow cytometry is currently done at multiple steps before, during, and after the process, and thus lead to a significant percentage of the entire analytical effort. In fact, analytics have become the major bottleneck after manufacturing challenges could be strongly mitigated due to closing and automating workflows, for example using the CliniMax Prodigy. It is therefore consistent that we now transfer the success of the automation and manufacturing also to the analytical space. Similar to the manufacturing procedures, the development of different analytical procedures, either for in-process controls, release testing, companion diagnostic, or concomitant research, are all built on decades of experience with cells and the demands of complex cell and gene therapy modalities. In the future, we will make even more sure that using manufacturing and analytical devices will come with the same look and feel, which will reduce the risk of errors and necessity for retrainings to a sheer minimum. Now, seeing is believing. I would like to present to you 2 slides which will prove how robust and scalable CAR T-cell manufacturing can be done on the CliniMax Prodigy. All analytical data were obviously generated on the MaxQuant platform using Miltenyi's in-house development antibody cocktails and express modes for data acquisition. What we compare with these graphs are two different sets of runs using the same 12-day TCT workflow setup on the Clinimax Prodigy, using quite variable healthy donor material. The first group of manufacturing runs were conducted in a PD environment. You can see them as they are marked in black color. The second group, marked with orange color, were conducted in a GMP-compliant cleanroom environment. The first two graphs from the left show very high comparability in expansion and viability of CD3 positive cells. The two graphs on the right show similar high comparability with respect to the amount and percentage of CD20 CAR transduced T cells. Taking this one step further, a couple of healthy donor leukapheresis were split before starting the manufacturing run and sent to two different manufacturing clean rooms. One is in Germany, you can see that with orange color, and the other is at the west coast of the US, depicted in purple color. Again, you can see that even with a fully different set of manufacturing personnel and local cleanroom environment and condition, the Clinimax Prodigy is able to lead to very comparable run performance with respect to T cell yield, viability, and CAR transduction efficiency. While you already know that the standard applications have quite some built-in flexibility, it will sometimes become necessary to build further customization of the application software on top of it. To address these needs, Miltenyi offers so-called customized application programming as a two-step service procedure. For proof of concept and/or process development studies, research use-only caps will help you to determine the optimal process flow, which can then be subsequently moved into a GMP-supportive cap, which is very specific and verified for your process of choice.
Hi, my name is Dirk Wingassen and I'm leading the analytical development team at our CDMO facility in San Jose, California. Miltenyi Bioindustry is a division of Miltenyi, offering CDMO services to our clients. These services comprise lentiviral vector production, process and analytical development, clinical cell manufacturing, and support and tech transfer services. We have experience operating in a global environment with 5 bioindustry sites represented across major geographies. We have manufactured greater than 700 clinical cell therapy batches and greater than 300 lentiviral vector batches worldwide. Our facilities with core competencies in clinical cell manufacturing are located in San Jose, California, Gaithersburg, Maryland, and in Bergisch Gladbach, Germany. Lentiviral vectors are manufactured in Gaithersburg and Teterow, Germany. Process development and tech transfers are performed at our facilities in San Jose, Bergisch Gladbach, and Shanghai, China. On this slide, I'd like to compare the early versus modern approaches to cell gene therapy GMP manufacturing. On the left side in gray, you can see typical unit operations needed to manufacture CAR T cells. In the middle in blue, an early manufacturing process is shown, which is comprised of many devices, whereas a modern process, shown in orange, limits the number of devices and in the best case only utilizes a single device such as the Clinimax Prodigy. This translates very differently to manufacturing facility layout, as you can see on the right side. The Clinimax cell factory is designed as a ballroom concept, taking advantage of integrated, automated, and closed processes. For example, cleanroom classes can be downgraded to C and D, whereas more open manufacturing processes still use Class A within B and C cleanrooms. Here we compare the different manufacturing strategies side by side. When assuming a production output of 2,000 doses per year, the Clinimax cell factory would utilize half of the manufacturing floor space, a quarter of manufacturing staff, and a third of the total number of cell processing instruments. Another distinct advantage of adopting the Clinimax Prodigy is seamless scaling. Utilizing the instrument early in development reduces risk for later stage manufacturing, as process optimizations would be easier to implement and site transfers are more reproducible when performed on the same instrument platforms. With Miltenyi's global support presence, trainings and instrument setup can be accomplished on shorter timelines, which is especially important when preparing for product licensing and commercial production. A decentralized manufacturing paradigm, which moves cell production to point-of-care centers, can also be accomplished using Clinimax cell factory layouts. The same design could be applied at multiple sites, and blueprints for training and quality control can be easily transferred to new sites. The traditional centralized approach takes advantage of processes that were developed for a single Clinimax cell factory unit by scaling out manufacturing to multiple cell factory units at a central manufacturing location. Here, manufacturing capacity can be rapidly increased because protocol and method transfers to new equipment are less complicated and relatively quick. Our CDMO services consist of a fully integrated end-to-end solution that consider every aspect of the client's journey from initial scope development to commercial manufacturing. Our strength is to apply existing technologies, a lot of them internally developed, but also flexibility to accommodate custom workflows. For example, we can customize processes on the Clinimax Prodigy using caps, co-develop processes and methods with our clients, and transfer in and out processes and methods at any point in time. We are able to reduce timelines quite significantly by leveraging existing processes and analytical methods, bringing the timeline for process development and tech transfer to GMP down to 4 to 8 months. Custom processes or methods may add some time. However, we have shown timelines of less than 12 months into the clinic are still feasible. I wanted to now switch gears to lentiviral manufacturing. Similar to cell therapy manufacturing, Miltenyi is at the forefront of current technology, for example, by using a third-generation lentiviral manufacturing platform that is considered very safe with no reports of any replication-competent viruses being detected after transduction. The use of a serum-free, scalable suspension culture is critical to allowing us to predict GMP manufacturing performance based on a 4-liter batch. This slide shows the path to commercial manufacturing. After producing research-grade vectors to screen for transduction efficiency or potency, clients usually decide to produce a 4-liter batch with their best candidate, which, based on our experience, would predict GMP batch performance. This would allow clients to skip engineering batches, eliminating process development efforts to scale up to larger size volume batches. For preclinical clients deciding to pursue GMP batches, we target around 11 to 13 months for the whole process from research to GMP grade vector, including manufacturing of clinical grade transfer plasmid as a predecessor to making GMP vector. Clinical clients who already produce GMP grade transfer plasmid can produce GMP vector after 4 to 5 months, followed by mandatory commercialization activities such as process characterization and PPQ campaigns. In summary, clients who choose Miltenyi Bioindustry can rely on a vast manufacturing experience, both for cell and lentiviral vectors. Best-in-class timelines can be achieved by utilizing existing cell therapy and lentivirus vector platforms. Flexible service offerings help accommodate most manufacturing processes. And last but not least, worldwide presence of bioindustry facilities ensures smooth tech transfer and expert knowledge on local regulatory requirements.
And with that, we are at the end of today's webinar. Thank you very much for joining us.